The principles of inheritance and variation. That Mendel laws of inheritance were completed, and now let us see how it is given in the textbook. Have you ever wondered why an elephant always gives birth only to a baby elephant, not some other animal? Or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents? Or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other, or sometimes even so different, or sometimes even so different? Wait. These are several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offspring. And inheritance is the process which a characters are passed. Characters are passed on from parent to the progeny. Characters are passed from parent to the progeny. It is the basis of heredity. Then variation. Variation is a degree by which a progeny differ from their parents. So parents, offspring, both they don't look like completely similar. They can't be like a clone. Some variations can be seen over. So genetics deals with this heredity and variation. Heredity and variation. Humans knew from as early as 8000 to that 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possess desirable characters. For example, through artificial selection and domestication from ancestral wild cows, we have well-known Indian breeds like Sahiwal cows in Punjab, which is produced. We must, however, recognize that enough our ancestors knew about the inheritance of characters and variation. They had very little idea about the scientific basis of this phenomena. Then comes the Mendel. Mendel laws of inheritance. So that it was during the mid 19th century, the headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on the garden peas for 7 years, 1856 to 1863, and proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size. That is very, very important to be remembered. Mendel did experiments had a large sampling size, which have greater credibility to the data that he collected. Okay, then also the confirmation of his inferences from experiments and successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposing tribes. Two opposing tribes. We call them as alleles, like tall or dwarf plant, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination that before we continue, let us see the contrasting characters of the Mendel. We have 7 contrasting characters. Among them, some we did already. So let us come with the known one. Let us come with the known one. We say we did this particular class. A tall plant and dwarf plant. This is the character is height, stem height, and allelic forms or contrasting traits are said to be tall and dwarf. That contrasting traits are tall and dwarf or allelic form. We use the word. Character is stem height. Next, flower position. For flower position, axial and terminal. That axial and terminal. The position of the flower is a character. So what are the allelic form? Axial and terminal. Flower position axial and terminal. Okay. Next, pod color is another character we have taken. That pod color green and yellow. Pod color is the character. Allelic forms are said to be green and yellow. Allelic forms are green and yellow. Next, pod shape. Pod shape, full and constricted. Character is the pod shape. The contrasting traits are said to be full and constricted. Next, flower color. All these are the characteristic features of pea plant only. Flower color. Character, flower color. The allelic forms or contrasting traits are violet and white. Okay, so then these we know already. That round and wrinkle seed shape is the character. Allelic form or contrasting traits are round and wrinkle. Next, seed color, yellow and green. Seed color, yellow and green. Seed color is the character. For seed color character, that there are two allelic forms, yellow color seed and green color seed. Okay, some of them we did already. Monohybrid cross, we take the example of stem height, tall and dwarf. For dihybrid cross, we have taken two characters, seed shape and seed color. Seed shape is round and wrinkle, and seed color yellow and green. So that to be remembered. Okay. Next, let's continue. Mendel conducted such artificial pollination or cross pollination experiment using several true breeding pea lines. Okay, then what I mean, that means you have to understand the text sentences after all listening to that Mendel experiment only. That means that if you are not so that when we crossed a tall plant with a dwarf plant, when we crossed a tall plant with a dwarf plant, pure tall plant, true breeding pea lines, that meaning for that. Because basically, in case of these particular plants, they have a continuous self-pollination. A true breeding line is one that having undergone continuous self-pollination. Means this is self-pollinating, tall plant is self-pollinating plant, dwarf plant is a self-pollinating plant. That's why we can announce these are true breeding pea lines. Okay, so that these kind of Mendel selected these kind of 14 true breeding pea plant varieties. 14 true breeding pea plant varieties. So that 14 on display you can see here now. That 14. That means that varieties like round, wrinkle, yellow, green, 7 characters and 14 allelic forms. That's the meaning. 7 characters. Each character is having 2 contrasting traits. In that way, it was said to be Mendel selected 14 true breeding pea plant varieties as pairs. That means 14 true breeding pea plants as varieties, 7 pairs of characters, or we can say 14 true breeding pea plant varieties as pairs which are similar except for one character with contrasting traits. That's what I mean. For height, tall and dwarf, for seed color, that yellow and green likewise. Okay. So some of the contrasting sites are selected for a smooth or wrinkled seed, yellow or green seed, inflated or constricted, the green or yellow parts, tall or dwarf plant, all these are the contrasting traits. Do remember there are 14 true breeding pea plant varieties like this. And again showing in the table, 7 characters, stem height, flower color, flower position, pod shape, pod color, seed color. So each one having 2 allelic forms. So that means total 14. Okay, so likewise. So this is a table to be remembered. Among these, for experiments, we have taken stem height as in monohybrid cross and seed shape and seed color in dihybrid cross. Okay, so next let's continue. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed a tall and a dwarf pea plant. to study the inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. What do you call this as? F1 generation. In our normal language, we say simply a tall plant crossed with dwarf plant. What you got in F1? All plants are tall. Mendel observed that all the F1 progeny plants were tall, like one of its parent. Which parent? Dominant parent. None were dwarf. None were dwarf. In F1, there is no dwarf. He made similar observations for other pairs of traits. He found that F1 always resembled either one of the parent, and that the trait of other parent was not seen in them. Like yellow-greener cross, only yellow appeared in F1. Round and wrinkler cross, only a rounder appeared in that way. Okay. So Mendel then self-pollinated the tall F1 plants and to his surprise found that in F2 generation, some of the offsprings were dwarf. That means, let me remind you the cross total again. A tall plant crossed with a dwarf plant. In F1 generation, all the plants are appeared tall. When F1 is self-pollinated, some of the plants, nearly 25% plants are dwarf appeared F2 in F2 generation. Okay, that's what we are talking. F1 generation was now expressed that let's read again. The proportion of the plants that were dwarf were 1/4 of the F2 plant. Here it is, we are talking 1/4 of the F2 plant, and the tall plants are and dwarf or traits are identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. That means no external morphological variation they show with their parents. We are talking about 3/4 tall plants are appeared, 1/4 dwarf plants are appeared. Phenotypically, they look like their parents. Okay, so then let's continue. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. None were of in between height. In between height, intermediate stages not came. That is very important to remember. Similar results were obtained with other traits that he studied, while like yellow and green, round and wrinkled. F1 generation, while at the F2 stage, both the traits were expressed in proportion of 3:1. That means you need to understand Mendel not only stopped at tall plant, all the 7 characters with 14 allelic contrasting forms. Allelic traits are contrasting traits he did, and in all the times he got that 3:1 ratio in F2 generation. Okay, so this is a cross we are showing tall and dwarf for cross, the artificial cross pollination. And then what you got in F1 all tall and F1 cross with F1 self pollination. In F2 we got 3/4 tall plant, 1/4 dwarf plant, or in other words 75% tall and 25% dwarf. Let us try to understand this in the form of that Mendel observation. Based on this observation, Mendel proposed that something was being stably passed down. He don't know actually. Do you remember Mendel don't know any a word like gene they are using actually, but Mendel don't know it actually. Because gene, the word proposed by Johansen, not Mendel. At the time of Mendel, he is the beginning of genetics. Okay, based on these observations, Mendel proposed that something was being stably passed down, unchanged, from parent to offspring through the gametes over successive generations. He called these things as factor. Nowadays, so-called gene, whatever we are saying, that is factor according to Mendel. Genes, now we call them as genes. Genes, therefore, are the units of inheritance. They contain. That means, according to us, capital D are genes. But Mendel doesn't use that word. For our understanding, we are using. That he said it as there are some factors are present transferring from parent to the next generation. So genes, therefore, are the units of inheritance. They contain information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as allele. Genes which code for a pair of contrasting traits are known as allele. That means tall allele, dwarf allele that I am talking about. Which they code for same character. Which character? Stem height. Okay. So if we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage. That's why we use the capital T. Then small alphabet for other types. For example, in case of the character of height, capital T is used for the tall trait and the small t used for the dwarf. Similarly, we use capital Y for yellow and small y for green. Capital R for round and small r for wrinkle. Hence, in plants, the pair of alleles for the height would be like this: homozygous tall, heterozygous tall, and dwarf. Mendel also proposed that in a true breeding tall or dwarf P variety of the allelic pair of genes for height are identical. If they are identical, he called it as homozygous, like capital T, small t, respectively. Okay, then that capital T and small t are called the genotype of the plant. While the descriptive terms we use simply tall and dwarf. If you see this is tall. Description we say. If their genotype will be either homozygous tall or heterozygous tall. That's what we are talking about. Okay, then what then would be the phenotype of a plant that has genotype T, capital T, small t? We are talking about that. That will be tall. As Mendel found the phenotype of the F1 heterozygote, capital T, small t, to be exactly like the small capital T parent. That means in genetical terms, heterozygous tall plant is exactly same like homozygous tall plant in expression. There is no height difference. That's what we are talking about. No height difference came in both. Both are like same. He proposed that in a pair of dissimilar factors, one dominates the other, as in the F1, and hence is called dominant. So this in our language, when we say one dominates the other, who dominates? Capital T is dominating the small t. That what do you say? The dominant factor, while other factor will be recessive. Here, in this case, capital T is the dominant one and the small t is the recessive one. Capital T is the dominant and small t is the recessive one. In this case, here it is shown that dominant over small t, that is recessive. He observed identical behavior for all the other characters, trait, pairs that he studied. It is convenient and logical to use the capital and lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. So that do not use capital T for tall and small d. We should not use like that. We have to take dominant character as a capital letter. Their small letter will be for recessive character. Dominant character for that dominant allelic form is the letter will take, and their small t will take. That to avoid the confusion we are talking about. You will find it difficult to remember capital T and D. So that's why we are using capital T, small t. Alleles of the same gene or character or not. Alleles can be similar as in the case of homozygote, like capital T, small t, or dissimilar in the case of heterozygote, capital T, small t. So that's what I'm talking about. Homozygous. And this is called heterozygous. So need to find out the difference between the homozygous and heterozygous. So since the heterozygous plant is, that means capital T, small t plant is heterozygous, and for genes controlling one character, height, it is monohybrid, and the cross between the capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by a process of meiosis, the alleles of the parental pair separate or segregate. Do remember this. We are talking about this particular, yesterday's law of segregation. So we are talking about this one. We are talking about capital T and small t. So when they segregate from each other, only one allele will be transmitted to the gamete, capital T, small t, like this. Only one allele will be transmitted. So at the time of segregation of alleles in a random process, there is a 50% off. Okay. So there is a 50% chance of a gamete containing either allele has been verified by the results of the crossings. In this way, gametes of the tall plant, the capital T plants have the alleles capital T, and the gametes of the dwarf small t have the allele small t. So let us correlate these. We are talking in the parental generation. Here the gamete, capital T, gamete, capital T. Here small t, small t. That means for every theoretical sentence, we have to give some representation. If some representation will come in the mind, that will be easier to you. Okay. So let's start. Let's continue. Capital T from one parent, say through the pollen, and small t from the other parent, then through the egg, and united to produce zygotes that have only one T allele and one small t allele. So talking about this. So crossed like this, that gametes were taken, similar kind of gamete, that's why I'm writing only one. One is the male gamete, other is the female gamete, both unite and form the zygote. So that, in other words, hybrid has capital T, small t. What do you say this as? Heterozygous tall. Okay. Since these hybrid contain alleles which express contrasting stripes, the plants are heterozygous. The production of gametes by the parents, the formation of the zygote, the F1 and F2 plants can be understood from a diagram. The Punnett square board, in the name of scientist Punnett, we are talking, as shown in figure 5.4. This is the diagram. Okay, then it was developed by a British genetist, Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetic cross. The possible gametes are written on two sides, usually a top row and left columns, right, and all possible combinations are represented in boxes below in the squares, which generate the square output form. I'll show you once here. This is the form. This is the form capital T, small t, capital T, small t. Will take the gametes like this in F2 generation. Capital T, capital T, capital T, small t, small t. Yeah, so this is the format. So much better format we are showing here, the Punnett square board. No need to draw these diagrams. That means when we see this, this is physical expression. When we see this, this is genotypic expression. At least you should understand in that particular form. Okay, so physical and genotypic expression. Let's talk about the Punnett square. Shows the parental tall, capital T, and dwarf, small t, female plant. The gametes produced by them and the F1, that heterozygous tall progeny. I hope you are understanding because we have completed total monohybrid and dihybrid class. The F1 plants of genotype are self-pollinated. The symbols. So the F1 plant of the genotype, capital T, small t, when self-pollinated, produces gametes of the genotype, capital T and small t, an equal proportion. When fertilization takes place, the pollen grains of the genotype, capital T, have a 50% chance to pollinate eggs of the genotype, capital T, as well as genotype, small t, also 50%. Also, pollen grains of the genotype, small t, have 50%, and eggs have genotype, capital T. Here the confusion comes. What he is saying, simply you should just try to understand. What he is saying, we are talking about F1 plant. Okay, self-pollination. One side male gametes, other side female gametes. So he is just talking about this. During meiosis, when the gametes are getting formed, that 50% of the gametes are that the pollen grains, pollen, and 50% pollens are dwarf, 50% pollen are having tall allele. In the same way, here also, the 50% are eggs are tall allele, 50% are with dwarf allele. Is saying where it all happened when we cross F1 with F1, self-pollination. Okay. So that means for every theoretical sentence, if you made such genotypical representation, don't try to understand with theory. You already should know. After that, after all doing experiment only, when it come, you'll come to know what you want to say exactly. So that's why I explain monohybrid cross, and now I'm reading how it is given in the textbook. Okay. So that capital T, capital T, capital T, small t, small t, small t. Where you got this? In F2 generation. That genotypically. Okay. So phenotypically we say 75% tall and 25% dwarf. So these are the plants appeared in F2 generation. Okay, so 3 tall plants and 1 dwarf plant. We got. So how do we say this? This we can say like this: 3/4 tall plants, 1/4 dwarf plant. 3/4 tall plant, 1/4 dwarf plant. Okay. Then in form of this is phenotypical expression. In genotypical expression, what we have to say like this, that F2 offsprings are appeared from the box I am drawing outside. So how we say 1/4 homozygous tall and 2/4 heterozygous tall and 1/4 homozygous bar. We should say like this. So here text will confuse you. So they give like this 1/2 capital T small t. That means 2/4 when you do like this, you'll get 1/2 and means I'm talking about this is 25% and this is 50%. That means what this is heterozygous tall 50% and these are 25%. Don't confuse by looking toward this particular paragraph. So this paragraph will make you, most people will confuse here. So that's what I am saying here. If you make a graphical representation and when you come here, they don't. And let's continue. Let's continue. So just for reading sake, I'm saying we know all this. From Punnett square, it is easily seen that 1/4 of the random fertilization led to capital D, homozygous tall. 1/2, that means 50% are heterozygous tall. 1/4, that means 25% are homozygous tall in F2. Through the F1s have a genotype capital T, small t, but the phenotypic character seen is tall. At F2, 3/4 of the plants are tall, where some of them are homozygous tall while others are heterozygous. Externally, it is not possible to distinguish between the plants with the genotype that homozygous and heterozygous. We can't say externally. Both look like same. That how they look like? They are tall plants, that's all. Simply we say they are tall plants. These two are tall plants, phenotypical. Hence, within the genotypic pair, that heterozygous tall, only one character, capital T tall, is expressed. We are talking about this. This is only expressed. Among this only expressed. Okay. What do you call that expressed one? Dominant. Capital T, tall is expressed. And the capital T of tall is said to dominate over the other allele, small t. Other allele, small t, which is the dwarf character. It is thus due to this dominance of one character over the all the F1 are tall through the genotype capital T, small t. And in F2, 3/4 of the plants are tall. 3/4 of the plants are tall through genotypically that 50% are heterozygous tall and 25% are homozygous tall. Just while reading, if you keep this in mind, F2 offsprings which brought out from the box, you can understand easily. So what I'm saying, these are 1/4, they say 25%, 1/4 homozygous tall, and 2/4, that means 50% heterozygous tall, or 1/2 we can say. Then these are 1/4, 25%. That we should understand. Phenotypically, will make the full box. Phenotypic means the total 3/4. Phenotypic, when you want to say physical expression wise, 3/4 are tall and 1/4 are dwarf. Okay. So then that genotypically, mathematically condensable in the form of the binomial expression. That in other ways they are trying to explain that here it is. Where we are here. That this leads to a phenotypic 3/4. We said 1/4 dwarf, 3:1, but genotypic ratio 1:2:1. Okay, so here don't confuse by looking this 1/4, 1/2, 1/4. Actually, it should be written like this: 1/4, 2/4, 1/4. 1/4 are capital T, homozygous tall. 2/4 are heterozygous tall, and these are here. That's homozygous dwarf are 1/4, 25%. Okay, so here they shown like this 1 by 2. They get just shown like this 1 by 2. So just try to understand. Okay, next. This particular ratio mathematically condensable to the form of a binomial expression ax plus by whole square that has the gametes bearing genes capital T or small t in equal frequency. So I will show you that also. When we are taking the gametes equal frequency, 50% tall gametes, 50% dwarf gametes came. That is one to say. The expression is explained as given below, that capital 50% tall gamete, 50% dwarf gamete. It's just like A plus B whole square. Okay, so what you get? 20 like this, the whole result we have to see the whole result. 25% expanded. A plus B whole square like expanded. I'll show you in that form. Capital T plus small t whole square. That means what he did, capital T small t into the plus capital T small t. In this way they made it up. Okay, so don't get confused. When mathematics comes, immediately problems will come. Here I am writing 1 by 2, 1 by 2. 50% of the gametes you want to see. So when the 50% of gametes are present, the ultimate result will be like this. Again, Malli same. Raise 1/4 homozygous tall 1, 2, 1. 1 is to 2 is to 1 ratio. Or that 1 by 4, 2 by 4, 1 by 4. Okay. So then it will be shown like that. 1 is to 2 is to 1. Or it will say like this. 1 by 4, 1 by 2, 1 by 4. Okay, so that also in the same format it will come. This is 25%, here 50%, 25%. Okay, so that here also 25%, 50%, and 25%. Only thing you have to stick on that these are homozygous tall, these are heterozygous tall, these are homozygous bar. Here also the same. These are homozygous tall, and these are heterozygous tall, and these are homozygous bar. He is talking about F2 generation aspect. So that's it. That's why I used to say here, let us learn our basics, whatever we learned in earlier classes. So after that only, when you see the textbook, you will understand. The Mendel self-pollinated the F2 plants and found that F2 plants are continued to generate dwarf plants in F3 and F4 also. He continued the experiment. We did F3 actually. If we concluded that genotype of the dwarf was homozygote, small t, small e, what do you think he would have got? They had a self-pollinated tall F2 plant? Yeah, he did, that F2 tall plant. F2 offsprings, when he self-pollinated, at the time also, he got dwarf plants. From the preceding paragraph, it is clear that through the genotypic ratios can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait. It is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2, if I take that capital T or small t, homozygous or heterozygous composition, that cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. Then he called it as a test cross. Sorry, F1, here it is. Tall plant from F1, F1. Here a small correction text here. F1 with dwarf plant, the dwarf parent. What do you call it as? Test cross. Okay, so let me show you from the beginning. It's a parental plant. When we cross that F1 plant, the parental plants each other, what we obtained in the F1 generation, heterozygous star. Okay, he did this cross. What do you call it as? Test cross. In a typical test cross, an organism P plants here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. That means test cross for what? To know the F1 plant is either dominant parent or recessive parent. You might have a doubt, I wrote already genotype, but you should face this kind of thing in garden. When you see this F1 plant, both homozygous, heterozygous look alike, isn't it? So that's why we do cross F1 with recessive parent. You should have imagined like this. A tall plant cross with a dwarf plant. In F1, all tall plants are appeared. We don't know what the genotypic configuration they have. That's why we cross with their recessive parent. We call test cross. The progenies of such cross can easily be analyzed to predict the genotype of the test organism. Means this one. So that genotype, the phenotype we know easily, but genotype, to know the genotype, we need to do the test cross. That to be understood. So results of the typical test cross where violet color flower is dominant over white color flower, that's shown in the figure 5.5. We go there and we see there. Okay, so we'll go there and we'll see that particular. We did this for Mendel, the dominant plants. Okay, so you see here. So here all flowers are violet. Small w, what we did. That homozygous recessive. Here you see the cross. We did a cross here, a test cross. See the diagram. Capital W, small w. Small w, small w. That means F1 heterozygote crossed with recessive parent. We got 50% violet, 50% white. So we predicted and we declared that unknown flower is heterozygous. Okay, unknown flower is heterozygous. In the same way, that here you see the cross. That a plant, that particular in this cross, we got all violet flower. Okay, so you will be better understand we are doing cross between always from with tall plant we are doing. So let me show you the tall plant like this. So what we get, all are tall plants, isn't it? All are tall plants. So what you understand, that given tall plant is homozygous dominant. Okay, so here, so you that I'm writing genotype, but you should understand we know we don't see these letters on the plant, isn't it? So when we do like this, capital T, small t, small t, small t, we get some 50% dwarf plants are appeared. So that unknown flower is heterozygous, we declared. Okay. So in that way, you have to go. That same example we have explained earlier, that is with the help of violet and white flowers they explained. Okay. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? That we did now only. Using the genotypes of this cross, can you give a general definition for test cross? Yeah. F1 cross with recessive parent is called test cross. It is to know the F1 plant either homozygous or heterozygous. So based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called principles or laws of inheritance. The first law or law of dominance and the second law is the law of segregation that we have seen in the previous class. Now just a formality, how it is given in the text we are looking forward. Law of dominance. The characters are controlled by discrete units called factors. The so-called factors, we call them as genes, isn't it? The factors occur in pairs. The factors occur in pairs. I'll show you lot of factors. We'll did till the time. All these factors, they are all occur in pair, isn't it? So in a dissimilar of factors, one remember of the pair dominates and other is recessive. Where it is dissimilar, here it is dissimilar. So in that one is dominant and one is recessive. Dissimilar, what I mean? Heterozygous. The law of dominance used to explain the expression of only one of the parental characters in monohybrid cross in F1 and the expression of both in the F. It also explains the proportion of 3:1 obtained at the F2. Yes, so do remember this law of dominance used to explain expression of only one of the parental characters in a monohybrid cross. Let me do again. So this is the appearance in F1. We got, so what we said, F1 tall plants are expressed. So I said declare tall is the dominant. Okay, and the expression of both in F2. We got in F2 along with the tall plants we obtained dwarf plant. It also explains the proportion of 3:1 obtained at the F2. Why 3:1 are appeared? We can say this is dominant, this is dominant, this is dominant. That's why they are appeared. Okay, so that's it. Law of segregation. This law is based on the fact that alleles do not show any blending. Blending, what I mean, influence, that the both characters are recovered as such in the F2 generation. So one of these is not seen in the F1 stage. So in F1, we got a tall plant. So and we declared according to law of dominance, capital T is the dominant one, small t is the recessive allele. Okay. So during this particular period of time, any that dominant that any influence is there on that particular small t? Is it changed? No, that's what we are talking about. That no change at all. So at present, it is not expressing. When time comes, without any change, it will go for their original expression. That you have to remember in law of segregation. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other. I am showing the segregation, separation from each other such that a gamete receive only one of the two factor. Each gamete have only one of the two factor. We have two factors here, capital T and small t. Then a pollen contain capital T. Then a pollen grain contains small t. Or egg contain capital T. Egg contains small t. The possibilities. At what time? Segregation. Now I am talking about this particular gamete expression. So this is recessive expression. This gamete express recessive. No influence of all is there now. So that's why we can say that gametes, when they get segregated, they retain their original character, the expression without blending. Of course, a homozygote parent produce all gametes that are similar. That we know. Okay, so when we come to the homozygote parent, but heterozygous one produce two kinds of gametes. This is the heterozygous one I am showing, the two kinds of gametes, each having one allele with equal proportion, 50% of tall gametes, 50% of dwarf gametes. Okay. I am trying to explain each and every sentence here. That's why it's taking little length. But I request all of you to read back again. Read back again. So that's it about that particular Mendel monohybrid cross. We continue with these particular issues, that incomplete dominance, codominance, Mendel dihybrid cross, and independent assortation of the Mendel. That all we have to see. That all we need to continue with.